Thank you. So the experiment that we're going to do today is called in your handout maybe serial dilutions and protein determination. There are actually three major names for the technique that we're going to use today. So you can call it protein determination. You'll hear that one. It can be called a total protein assay, or it can be called a Bradford assay. And these really are all the same type of thing, and they're alternative names that may be used. So when there are major alternative names for a procedure or a process, I want you to know all of them, so that the next graduate professor that you have, if they start talking about a Bradford assay, you're like, no, never did that. And yes, we did that. You've done that. And all of these have in common, they are all a particular name for a process that you can use to determine the amount of total protein, that's the amount of total protein in a solution. All of our labs that we do in cell biology have to do with protein experiments. So one of the first basic techniques is to know how much protein is in this sample. Those of you that had biochem, I knew you had boneless chicken, but you had a process where you took chicken, you ground it up, you got a protein extract, and then it would be useful to determine, okay, in this tube, here's my tube, it's got this clear extract. How much protein is in there? All the protein together, mixed together in solution. What is the concentration of total protein? People in biochem, what did the solution look like when you got to that point?
It's really cloudy.
It's cloudy. What color is it?
Like pink.
Pink?
Ours
From the myoglobin? That's the wrong answer. You're supposed to say clear.
Oh.
Okay, so no, it was pink. A lot of times a total protein extract, depending on how much purification you put it through, is going to be clear. Because most proteins don't have a color to them except for myoglobin, hemoglobin, and so on. So it's clear, and we want to know how much protein is in that solution. The way that we're going to determine that is to add a particular dye to the protein mixture, which is called Bradford reagent. Bradford reagent. And Bradford reagent in its natural state is brown. So it's brown when it's uncomplexed, brownish color. But when it is complexed with protein, it is a blue color. So it's blue when it is complexed or it is bound to protein. Binds protein residues, and in the bound state, it's blue. So the more blue the mixture is, the more protein it contains. So more brownness means more uncomplexed, more not bound to protein. More blueness indicates more protein. You can do this lab. If any of you who watch the video, you're ever going to be high school teachers, or you teach in a place with very limited material resources, you can actually measure the blueness with the naked eye, cheaply. If you've got the technology available, you can measure the blueness via a spectrophotometer, and that's what we're going to do in this lab. We're going to measure, we're not just going to say, oh, this one's more blue, this one's less blue. We're going to measure how much protein is there by using a spectrophotometer. Probably all of you have used a spectrophotometer in chemistry before, and recall that you have a light bulb, not a chicken leg, but a light bulb this time that is shining light through a sample. And the amount of light that goes in can be compared with the machine by the light that comes out. So you've got the amount of light in, the amount of light out. If a lot more light goes in than out, there was a lot of light absorbed. So we're going to be measuring the absorbance of light shining through our sample, which is a complex of Bradford reagent and a protein mixture. Very important for this experiment that the wavelength of light that we're putting in is particularly 595 nanometer light. There are different light settings if you're going to assay DNA, RNA in a spectrophotometer, but for protein, the complex of Bradford plus protein absorbs best at 595 nanometers. That's important. So if you ever go on and do this in the future and you look back at this notebook, it's really important what the setting is. So you can use this method to figure out how much protein is in solution. And if you want to know exactly how much protein is in your solution, you're going to need concentration standards. You really need concentration standards, or you need known concentrations of protein to make a standard curve. Yes, standard curve. It's not really that bad. Okay, so if I have a graph like this, and I'm going to plot concentration on this axis and absorbance on this axis, what do I expect to happen to the absorbance as the concentration increases? Increase. As the concentration of protein increases, the blueness will increase. And the absorbance will increase. The transmittance will decrease, but we're measuring absorbance. So we're going to have a relationship that is like this. If you didn't already draw that line, I'm going to draw a different line. So concentration versus absorbance is really a curve that's shaped more like that, if you look across a wide range of concentrations. But there is a certain range of protein concentrations that's basically linear for concentration over absorbance at that range. And that is, in most assays, between 125 to 1,000 micrograms per mil. 125 to 1,000 micrograms per mil. That can be a little bit different for different proteins, but that's generally the range where you can get a linear relationship between those. And if you go on and do further research, you need to remember that. So if you're trying to measure concentration of a protein and you think it's in the 5 mg per mil range or 5,000 microgram per mil range, you'll need to dilute it to get a good result. So what we're going to do is each group, I'm going to give you a tube of protein that has a concentration that says on the tube that it's 2 mg per mil. 2 milligrams per milliliter, or 2,000 micrograms per microliter. That's what you're getting. And I want you to make 4 dilutions. 4 dilutions from that fall within this range. And this is the first year. I'm going to let you decide what you want the dilutions to be. You'd be wise to keep it within this range. So you're going to make 4 dilutions of known concentration. Should you use the undiluted tube for this assay?
No.
You should not, because that's outside the linear range. So pick 4 dilutions, make 4 dilutions, and then I want you to get their absorbance values in triplicate. So what do I mean by that? So let's say that you decide that you're going to check dilutions. I'm going to make up ones that you definitely will not use. You're going to make dilutions of 173 micrograms per mil. That's going to be your first one. 209, 712, and then 967. If you want to use those, you can. It's going to be a tough dilution. So if those are your standards, make up those dilutions, and then from each of those, you're going to make 3 samples. So three samples, and to make these, you're going to put 40 microliters of the sample plus, this is lame, 1,960, great, this is on video for years, of Bradford assay. Bradford assay dye. For a total volume of what? 2,000 or 2 mils. You need 3 of those because you're going to assay 3 times. So you really need, I would advise you to make up a tube of this dilution that is 6 mils total. You need 6 mils, really you need like 8 mils in case you spill. That's a good rule of thumb. 8 mils total, or 10 if 10 makes it easier. And then you're going to take 3 replicates of each dilution, and you're going to do an assay tube for that. So how many assay tubes are you really going to have? 3 per dilution. 12. You're doing fine. You're doing really well to not have any material on this lab to this point. So you're going to have 12 tubes that you're going to assay. 3 of these, 3 of these, 3 of these, 3 of these. And each individual tube is going to have 2 mils. But you need to make 8 or 10 of the overall dilution. Does that make sense? That makes sense, okay. And then you're going to, once you have those dilutions made, you're going to put them in cuvettes. And cuvettes hold the sample in the correct orientation for the light beam to pass through for the spectrophotometer. And you need 2 mils, you need more than 1 mil, so 2 mil is a volume that will raise the liquid up over the light beam. You're going to measure the absorbance. And so you'll get absorbances for these. You'll have 3 samples of this, 3 samples of this, 3 samples of this, 3 of this. And you're going to average those. You'll get the average absorbance for that dilution. You'll get the average absorbance for this dilution, this one, this one. And then what you're going to do later on, when you have recorded all of these values, is you're going to plot concentration on this axis, absorbance over here, and you'll take those 4 averages, and what you hope is that they form a reasonable looking linear regression there. And actually this works pretty well if you're careful. This is some satisfying data, unless something weird happens. Then you're going to plot a best fit line. It needs to go through 0, 0. This will actually be a blank. Need to add that onto the list. So you'll have a blank that has 0 mgs per mil, micrograms per mil, and you will use that to 0 the machine. You set that to say, this has 0 protein. I say, this is 0 absorbance. And that will become this point. So you'll have 5 points, and you do a best fit line through those. Now when you're doing this, you can do a graph by hand on graph paper, or you can do a graph in Excel. But one of the things that you're going to do on the lab exam is to do one by hand in the lab with samples that I've set up for you. So you don't have to do the dilutions, but you do have to be able to show that you can do a reasonable regression standard curve by hand within like 3 hours. I mean, you got to do the whole test within 3 hours. But do a best fit line. How do you do a best fit line by hand? You done this? Is it from the Stone Age?
Draw a line.
You just draw it?
Just draw a line.
Just like that.
Yeah.
A roller.
A ruler? Is there a rule about how you do such a thing? There's one other thing to know.
Two ends, make a straight line.
So it needs to go through 0, and it needs to have roughly the same amount of dots on each side. And you should do it with a ruler, not like this. Unless you're like a great straight line drawer. Needs to go through, like if it'll have two dots on this side, 2 dots on this side. So that's generally how you do that. You need to make sure if you're doing a graph by hand that you've got the axis numbers appropriately spread apart, and that if you're using like 100 microgram tally marks, that they're all the same distance apart and so on. So that's going to be your standard curve. And then once you have that, then you can plot onto that the unknown concentration. So while you all are working on that part, I'm going to give you, or I'm going to make the unknown concentration and give you a sample of that. And you will treat it in the same way. You'll take this and you create a big 10 or 8 mil mix in these proportions, divvy it out into 2 mil aliquots. You know that word aliquot? A subdivision of the big. This is so important for this class. I'm glad I remembered this. Okay, if I have a big tube of protein mix. It's got 10 mils. If I take out 2 mils of that to do something with it, I call that an aliquot. It's like I sample a part of the whole. I'm going to say that word all the time. I think I said it for a whole semester one time, and people are like, I don't know what an aliquot is. Thank you for telling me now in November. That's a useful word. It's kind of like a sample, but it's a sample where there's some remaining in the larger mix. So, sample. An aliquot. So you're going to make this in the same way that you made these. You just don't know what the concentration is. But you'll get an average absorbance for that too. And then you can find, you know what the average absorbance is for the unknown. And you can plot it onto this line. So one thing that students in the past have done a lot is put try to put the unknown dot in here, which doesn't work. You can't, but they try to do it anyway. I don't know quite how, but the standard curve is from the knowns. The unknown you plot onto the standard curve. So if its absorbance is 0.4 or something like that, come over to where 0.4 is, extrapolate down, And then you can say, oh, okay, that's a concentration of 350 micrograms per mil, or something like that. Does that make sense? Have you done a thing like this before? And standard curves are a standard technique.
We attempted to do it.
You
never
attempted.
actually happened.
It never happened. It works pretty well in this class. I think because I tell you what absorbance to start with, and like that saves time, and that is the protein, the standard protein absorbance. So that's what you're going to do. The point of this lab is to find the concentration of an unknown protein. Yes, you're going to learn how to do a Bradford assay, but I've had some really great lab reports in the past that did everything amazing and then did not tell me what the concentration of the unknown protein is, which is the point of this exercise. So, yeah. Please put that, draw a box around it if you feel like it.
So how exactly are we supposed to get those 4 different dilution numbers?
The dilution numbers, I'm going to have you work that out in your group. So decide, you and your group decide, okay, what are our 4 dilutions going to be? And then work on how to accomplish that. So I'm going to give you one example. I'm going to give you an example. You're starting with 2 mg per mil, and if you decide to make one of your dilutions 500 micrograms per mil, I'm going to erase this now, okay? If you decide to make one of your dilutions 500 micrograms per mil, how would you do that? What's the approach? It's a very good question, by the way. But there's a reason I'm not spelling it out for you so much.
To convert it, 2 milligrams to micrograms?
That would help in the start. So you're starting with 2,000 micrograms per mil. What I need to get is 500 micrograms per mil. How much of this do I really need of that mixture? 10? Like 10's an easy number. So I need 500 micrograms per mil, 10 mils of it. How much of this do I need to use to accomplish that? I want 10 mils of that. How much of this do I need to put into a tube and then make up the difference to 10 mils?
4, no.
The number 4 is involved, let's
40.
say. The number 4 is involved. So this is 4 times more concentrated than this, right?
Yeah.
This is 4 times more concentrated than this. I need a fourth of this volume to be this.
Yeah.
Does that conceptually make sense? So I need a fourth of this volume to be this. There's this handy equation, V1C1 equals V2C2. Super simple. So what you want to do is figure out, okay, the final volume is this, the final concentration that I need is this is my starting volume. Got to get the units the same, but what would that volume be? So to solve this, what I'm trying to find is V1, right? So V1, V2, C2 over C1. So I have 500 micrograms per mil times 10 mils over 2,000 micrograms per mil. Cancel, cancel. 5,000 over 2,000. 5 over 2 should be 2.5 mil, is that right? Which is a fourth of this. Did you use this in biochem?
Yeah.
Yeah. A lot, all the time. Probably in chemistry.
In general.
So long ago. You know what happened? When I started teaching this class, I forgot about that equation. And I was like, I know how to do this, but I forgot the simple equation. And then someone, Dr. Vaughn mentioned it. I was like, oh yeah, that would help teach them. That would be a good idea. Because I forgot the equation, but I remembered the concept. Well, it needs to be a fourth of this. That's how you'll do it. The thing that you need to watch out for is that we don't have unlimited amounts of this. We don't really have unlimited amounts of this. So when you need to make let me go back to that part. You need 40 microliters of sample plus 1960 microliters of dye. The way that you probably should do this, because we don't have a lot of sample, is to make 40 plus 40 plus 40 for your 3 samples, your 3 replicates. That's 120 microliters. So you should aim to make 200 microliters of each dilution. And then once you get that, put in the tube 40, 40, 40, then add in the bulk dye. See what I'm saying? Here's what that's going to look like. So you need to make 200 microliters of the dilutions that you pick. So if you pick 800 micrograms per mil instead of 500, you're going to need 200 microliters of that. Here's the great thing about this. Let's change this equation. I know I'm going on for a long time. This is going to help you through the semester if you're able to do this pretty quickly. So say that I want 800 micrograms per mil and I need 200 microliters. I need 200 microliters. Do I need to change all this stuff to microliters? No. This is great. You could, but you don't have to because these are going to cancel when you do the division. That's great. That's going to save you time. If I've got microliters over here, I can go ahead and solve for microliters here. Do that now. Try that math. I'm going to do it over here. You can get super imaginary bonus points if you don't need a calculator. Not for real, but should be 80. Right? That's how you'll figure out your dilutions, but you decide. So every team is going to get a tube of this with a certain volume, and then you all decide how are you going to use that to set up your 4 dilutions. Questions? We've got 2 hours. It doesn't really take 2 hours, but you know, be organized, be careful, set it up correctly in the beginning. What you want to avoid is having to do the whole thing twice, and there's usually one team a year that has to do it twice. I don't take off for that, it's just like, oh man, we gotta do this again. So. Okay, and if you, I think